Presented by Caltech.
Thank you very much. It's my great pleasure to introduce tonight's speaker, my colleague, Mikhail Shapiro. Let me first give you a little background about Mikhail. He's an assistant professor of chemical engineering, a Schlinger scholar, and a heritage principal investigator here at Caltech. Mikhail received his PhD in biological engineering at MIT. Prior to that, he received his bachelor's in neuroscience from Brown University. Before he joined us at Caltech, he spent a few years in industry in a venture capital firm, but the lure of research called to him. He spent a postdoc at the University of Chicago and was a Miller Fellow at the University of California, Berkeley. His laboratory develops molecular technologies, allowing cells to be imaged and controlled inside the body, which we'll hear about a bit tonight. He's received a number of awards. I'll just list a few. He's a Burroughs Wellcome Career Award, the DARPA Young Faculty Award, the Pew Scholarship, the Sontag Foundation Distinguished Scientist Award, a Packard Fellowship, the Van Ness Award in Chemical Engineering, the Camille Dreyfus Teacher Scholar Award, and Technology Review TR35 Award for Innovators under the age of 35. And this year, this lecture is the Biederbach Memorial Lecture, given an honor for an assistant professor at Caltech to present a Watson lecture. So without further ado, please join me in welcoming Mikhail Shapiro for his lecture entitled Talking to Cells. Mikhail?
All right. Well, thank you, John, for that very kind introduction. And he actually read all of my awards, not just some of them. Yeah, thank you to the Watson Lecture Committee for inviting me. It's really a big honor to be here. And thank you all for coming. It's a distinct pleasure to have so many friends and colleagues here, and also people I don't know but who look very nice as well. And I'm delighted to share with you some of the science that we're doing in my lab. But first, I'd like to start by talking about the movies. And in particular, there's this movie that came out in 1966 called The Fantastic Voyage. Who here has seen it? Okay, all right, I see some fans here, okay. So for those of you who haven't seen it, the plot of this movie is that there's a nuclear physicist who has a secret equation in his head, and there's an assassination attempt by the Russians, and as a result of which he has a blood clot in his brain. And if that blood clot doesn't get fixed within 2 hours, he's going to die, and the Soviet Union is going to destroy America. And we don't want that to happen. And the issue is that the blood clot is deep in the brain, so surgery is not really an option. And so what happens in this movie is that a group of doctors and engineers go into a submarine that gets shrunken down by a millionfold, by 6 orders of magnitude, to where that submarine and its occupants are now the size of a red blood cell. And the submarine gets injected into the bloodstream of the scientist and courses through the blood, gets to the blood clot, fixes it, and the world is saved. Now, this movie has really inspired a generation of scientists and engineers to envision this as the real future of medicine. Because compared to drugs, which we take, which go everywhere in our body and cause side effects, and compared to surgery, where you have to cut somebody open to get in there, this is a very elegant solution, right? Because you can have something, this idea that you have little micro or nanoscale robots that are going to be roaming around in our bodies and acting as doctors. And so in my lab, we're totally bought into this idea that this is a future of medicine, with the exception that instead of this submarine being a mechanical object that gets shrunken down to the size of a cell, that instead it's going to be cells themselves, okay? Where we're going to engineer these cells to act as doctors inside of our bodies. And the reason for this is that if you think about what we want these microscale robots to do, cells already do 90% of it. Cells can migrate through our bloodstream to sites of disease. They can sense molecular signals in their environment. Based on those signals, they can perform computations and logic operations and make decisions. And then based on those decisions, they can take action. So a cell can synthesize and release a protein that can act as a drug locally, just at that right place under the right conditions. It can also kill other cells. So immune cells can attack another cell and kill it. It's also a therapy that can make more of itself, because cells can proliferate. And ultimately, when they're no longer necessary, they can self-destruct. Okay? And so the idea that has really emerged over the last 10, 15 years that we and other people are pursuing is that there's a variety of different kind of cells that we can co-opt where they do 90% of what we want, and now we're going to genetically engineer them so they have this additional 10% that does our bidding. Okay, and a few examples of that you may have heard of include regenerative medicine, where people have talked about stem cells that you might be able to inject into the brain that would replace dying neurons, or into the heart to treat heart failure. Immunotherapy, which is a really exciting area over the last few years, where immune cells can fight cancer. Okay? And unfortunately, cancer can be good at hiding from the immune system. And so the idea from this engineered immunotherapy is that you can take out a patient's immune cells and you can genetically modify them so they have a receptor on them that if you inject them back into the patient's body, they can go and find and identify the tumor and kill it. Okay, so you're giving them this extra capability. And the first 2 immunotherapies were approved by the FDA just within the last year for cancer. And then probiotics. Each of us has trillions of bacteria living in and on our body. And that's a good thing, because they're helping us. They're part of our natural homeostasis. But now you can take those bacteria and you can give them additional functionalities. You can engineer them, for example, that you swallow them. They'll go through your gastrointestinal tract, detect inflammation, and release anti-inflammatory compounds. And perhaps these are things that you've read about or seen in some of these magazines and in the scientific press and the popular press. It's really captured a lot of people's imagination for a way forward for medicine. However, these very sophisticated agents that are going to be injected into our bodies have a real limitation, which is that after I inject these cells into you, today I have no good way to tell where are they. Did they get to the right place? Are they alive? Are they carrying out the functions that we programmed into them? And we also don't have good ways to tell these cells what to do based on their location. And we think this is really important. And again, this was something that was presaged by the Fantastic Voyage, where that little submarine inside the body had this entire control room of people that were communicating with it via the radio and saying, turn right, turn left, where are you, et cetera. And so we'd like to communicate with cells that we've put into somebody's body. And the reason this is very challenging today is because most of the tools that we have for imaging and controlling the function of cells work really well under the microscope. And they're used by biology labs all over the world, including in our lab. And they're things that are based on these proteins called fluorescent proteins, which can interact with light. Okay, so this is a protein that I'll say a little bit more about, where if you take the gene sequence that encodes this protein and put it into a cell, now you can see the function of that cell under an optical microscope. Okay? And this gives you the power to visualize bacteria, animal cells, neurons, et cetera. And it's really powerful. But there's a big limitation in that light doesn't penetrate very deeply into our tissues. Right? If I try to shine this laser pointer through my hand, it's not going to make it through. Okay? And light gets absorbed and scattered within about 1 millimeter of entering tissue. And so it doesn't scale very well to large organisms. On the other hand, we have technologies like magnetic resonance imaging and ultrasound that use penetrant forms of energy. So magnetic fields and sound waves, by and large, have no difficulty getting into our bodies, which is why we can get beautiful images like this one of a fetus or this one of the human brain. But today, these technologies and these forms of energy are not connected to what happens inside of cells. Okay? And so what we try to do in my lab is to take the physics of sound waves and magnetic fields and these technologies and extend their capabilities down to the molecular and cellular level through biomolecular engineering, where we engineer and we evolve proteins or other kinds of things that a cell can make that have the right physical properties to interact with the sound wave or interact with the magnetic field in such a way that would allow us to see and control cells in the body. Now, my laboratory works both with sound waves for ultrasound and with magnetism for MRI, and I see several members of my laboratory here. So today, I'm going to focus on our work with ultrasound, and I apologize to the 6 of you guys who work on magnetism, just in the interest of time. But if you're interested, you can learn about the magnetic things later on as well. So I'd like to tell you about primarily 2 ideas. So one is we have a cell inside of our body, and let's say it's a microbe that's in our gut that we've put there for some beneficial reason. We want to be able to image it, and we want to be able to control it. I'm going to tell you how we can do both of these things. So for imaging, we're going to use ultrasound. It's the most widely used biomedical imaging modality in the world. Virtually every doctor's office has one of them. It works on a pretty simple set of principles. It's sound waves, just like the ones with which I'm communicating with you right now, that are transmitted into a tissue, and they move along until they encounter an interface with something that has a different density or stiffness, such as a bone, and then some of that sound wave gets reflected back. And because we know the speed at which the sound travels through tissue, based on how long it took between when we emitted it and when it came back, we can tell how deep this object is. Okay, and then we can scan our sound wave left and right, to and fro, and get an image like this one, which I'm pretty sure everybody here has seen in, and many of you have been in at some point in your lives. Now, ultrasound is probably most widely known for these kind of images, which might not look so impressive, but actually it's a very high-tech technology. And a beautiful example of this recently came from our collaborators in Paris, where what you're looking at here is an entire brain of a rat, which is several centimeters in size. And what you're seeing with all these little lines are individual micro blood vessels within this entire brain that are resolved at 7-micron resolution, so very small, and you're able to see it throughout the entire subject. And ultrasound is also very accessible. So when we think about making this widely available in the developing world, in all the different hospitals and neighbors in the US, now there are these ultrasound systems where all of the technology is just in this handheld transducer that you can plug into your Android phone. And there's another version that works with Apple, if you want. So it's something that could be widely disseminated. So what I showed you so far with the ultrasound is just looking at anatomy. But we'd actually like to go beyond that, right? We ultimately want to image cells. So what's available today are the equivalent for ultrasound of chemical dyes. Okay, so there's things called microbubbles, which as the name implies are micron-sized bubbles of gas that are encapsulated by some kind of lipid shell usually. And when these bubbles get injected into your bloodstream, okay, because they're of lower density, they scatter sound waves. So this allows doctors to visualize your blood vessels and blood flow in a very nice way. And in fact, this image of the rat brain is facilitated by the fact that you have these little microbubbles that are floating around in all those vessels. And so a question we asked a few years ago is, could a cell make something like this? Okay? And the inspiration just behind this question comes back to fluorescent proteins, where the way those originated is that people knew for a long time that certain kinds of organic molecules that we can synthesize in the lab are fluorescent, meaning that if you shine light at them at a particular wavelength, they will themselves return light at other wavelengths. Okay? And there was a discovery that jellyfish also fluoresce, and that they contain this protein that was named the green fluorescent protein that allows them to do this, because this protein itself also has a similar organic chromophore that's inside of this protein. And if you take the genes encoding this protein from the jellyfish and move them into bacteria or into other kinds of cells, then you can have those cells produce fluorescence. Okay? And this is what's really revolutionized biology. And so the question is, just like the physical properties of this organic dye were identified in something that a jellyfish could make, can the physical properties of a bubble be identified in some living creature that could help us do what we want? And the answer came from these species here. So what we're looking at are the South Bay salt ponds that you might have seen if you're landing in San Francisco International Airport. And the color in these bodies of water comes from photosynthetic microbes that are living there, so they're using sunlight for energy. And they had to solve an evolutionary challenge of needing to regulate their buoyancy. Okay? Because if they're floating all the way at the top of the water on a sunny day, they get burned. And if they're too low underneath the surface on a cloudy day, they don't get enough light for photosynthesis. So they want to be just at the right level. And the way they evolved to do that is by forming these really beautiful protein nanostructures, so things that are even smaller than a cell, inside of the cell, that are called gas vesicles, but for those of you who know this term, it's a misnomer. There's no lipids. It's a protein-shelled structure. And what's unique about this self-assembled protein structure inside the cell is that it's hollow on the inside and filled with gas. And the gas here is whatever gas is present in the surrounding cell will partition in and out, just like gas is partitioning at an air-water interface in my water bottle here. And what's remarkable is that the structure can be made completely in the cell based on the genetic program. Okay, there's a set of genes that encode proteins that find each other and assemble into this nanoscale structure. And because it's full of air, when the cell wants to float higher, it can just make more of these things, reduce its density and float higher in the water. And when the cell wants to sink, it can break these things down with proteases, proteins that chew up other proteins, and sink back down. Okay? So it's a really beautiful piece of evolved machinery that was discovered originally more than 100 years ago. And I just want to show you a zoomed-in picture of these guys. I find it really remarkable that the cells can make this. Because if you ask me as a chemical engineer, or if you ask other people here who are material scientists or mechies, to make you a nanoscale compartment that contains gas, is physically stable, and has a shell whose thickness is only 2 nanometers thick, right? We're talking about tens of atoms in thickness, and is physically stable and can even withstand several atmospheres of pressure. I would say that's going to be very difficult, and you have to give me a $20 million grant even to try it. But cells figured it out through evolution. And this is a picture of them intact here, so you can see the cell makes them very nicely and homogeneously. And then we can also break these things if we apply enough pressure, and this is what they look like under electron microscopy when they're broken. So you can see that it's just this really thin shell here. And so the question that we asked a few years ago is, what happens if we stick these things into an ultrasound? And we do a lot of experiments in my lab where we purify these proteins, or now we're taking live cells, and we're sticking them into some gel, and then we're taking ultrasound images. And so these were the first images we took of these proteins that were purified under ultrasound. And what you're seeing here with this color is ultrasound contrast. And this was exciting because this was the first time that a protein, something that a cell can make, could be seen with this imaging modality. And of course, after we saw this in vitro, one of the first things we said, let's inject it into a mouse and see what it looks like. And if you're injecting nanostructures that are of this size, and they're not functionalized to go anywhere in particular in the body, they're going to accumulate in the liver, because it's part of the liver's job to clear out nanoparticles from blood. And so what we're going to look at here is this outlined organ here is the liver of the mouse. I'm going to start the video, and we're doing an injection into the bloodstream, and you're going to see this liver go from being dark to accumulating brightness. Okay, let me just play that one more time. So it's going to start now. Okay, so it's dark. We start the injection, and then the liver starts turning bright. And this was the first time that we could see a protein with ultrasound in a living, breathing animal. Now, here we're cheating a little bit because we know when we're injecting it, so we know what to look for. If I just showed you an image like this one, how would you know if the contrast is coming from these gas vesicles or from something else that's going on? So here we can take advantage of the fact that we can collapse these things with pressure. And when they collapse, their ultrasound signals should disappear. So all we have to do is, if we have an image like this, is just crank up our ultrasound a little bit. Bam, and the signal gets erased. Okay, and because that happened at a particular pressure, we know that it had to have come from our gas vesicles. So this was really exciting. This, the first acoustic biomolecule. And in our lab, we take a lot of inspiration from the history of fluorescent proteins, where after it was first discovered from jellyfish, there were all these basic questions about green fluorescent protein, like, why is it green? How do you make it blue? How do you make it red? What is the chemistry and biology by which it forms in different kinds of cellular environments? And from investing in these basic questions, we got all these beautiful things like multicolor fluorescent imaging, or functional sensors where you can follow the dynamic activity inside of a cell, or even actuation where you can shine light and control these proteins. And because of this huge, versatile range of things that GFP can do, its developers were awarded the 2008 Nobel Prize in Chemistry. And we'd love to follow in their footsteps, not for the Nobel Prize, but for doing for ultrasound with our acoustic biomolecules what the fluorescent biomolecules have done for optical imaging. And over the last few years, we've undertaken a lot of different efforts in this area, which I don't have time to go into all of them, but I wanted to give you a few examples just to give you a flavor. So one of the first things we wanted to do was to establish a connection between the genetic and protein sequence that makes up these protein structures and their physical properties and how they affect the ultrasound signal. Okay? So we go in and we look at this set of genes that encode the structure, and we start messing around with them. And the first gene that we started to play with this protein is called GVPC, or gas vesicle protein C. That it's not the main protein that makes up the shell, so that one is called GVPA, but it's the second protein that sits on the outside of the shell and acts as a stiffening rod. Okay, just like reinforcement rods in an airplane or in a submarine. And we had a convenient way to play with this protein because we could purify these gas vesicles from cyanobacteria in our lab. Then we could chemically treat them to strip off their native GVPC. And then we can make new versions of this protein recombinantly in E. coli, meaning that we can manipulate their genetic sequence and make new versions of this protein and put it back onto these gas vesicles. So we have a very modular way to swap these things in and out, like Lego pieces. And we discovered some very interesting things. So one example of that relates to harmonics. So just like strings on a guitar or a violin, sound waves that scatter off of objects can also have harmonic signals. And in the case of the gas vesicles, we found that when we take away the stiffening protein from the outside, our structures go from producing just this fundamental frequency. So here we're transmitting ultrasound at this particular frequency here. And normally you just see that same frequency coming back at you. But if you make these things less stiff, now they start to produce harmonics. So here we're seeing double that frequency. And to try to understand this, we teamed up with some folks in our aeronautics department and did finite element modeling of these gas vesicle structures and discovered that these harmonics are coming from these buckling interactions. So from the point of view of this nanostructure, it's nanometers in size. The sound wave is tens of microns or hundreds of microns in size. From the point of view of the structure, a sound wave coming through is just like pressure going up and down, up and down. And when the pressure reaches a certain point, the structure buckles. Just like my water bottle, after the pressure gets high enough, it buckles. And it does so repeatedly with every cycle of ultrasound, millions and millions of times. And this buckling is a nonlinear process that naturally produces these harmonics. Now, the reason we care about these things is because we can take advantage of them when we do imaging. Okay? And the logic is that we know that there's a certain pressure below which they don't buckle and a pressure above which they do. So we can create an ultrasound imaging sequence where we can transmit a pulse at the full pressure that will cause them to buckle, and then we can transmit two pulses that are at half the pressure, okay, where it doesn't buckle. Now, for things that are scattering linearly that respond the same regardless of pressure, you're going to have one, and then you're going to subtract these two, so you're going to have 1 minus 1/2 minus 1/2 equals 0. Whereas for our nonlinear scatterers, these gas vesicles that are buckling, you're going to have 1 minus less than 1/2 minus less than 1/2 and get some signal back. And so indeed, when we implement this, if we do just plain old ultrasound imaging, and we compare our harmonic gas vesicles to some linear materials, they look the same. But when we implement this amplitude modulation sequence, now we see signals specifically from our harmonic gas vesicles that we've genetically engineered to allow this to happen. And this allows us to distinguish them for more complex background media, either artificial, as shown here, or in the body. So here we've deliberately put a pretty low concentration of these gas vesicles into the colon of a mouse. Under normal imaging, you can barely see it, but now under amplitude modulation, we see this nice bright signal that's coming from where they're located. Another example is multicolor imaging, right? So we talked about with fluorescent proteins how we have this entire beautiful rainbow that's been developed, and we'd like to do something similar with ultrasound. And what we discovered is that depending on which version of this GVPC protein we have sitting on the outside, we have gas vesicles that look the same under electron microscopy, but require different amounts of pressure to get them to collapse. Okay, so here we have 3 different versions that require 3 different amounts of pressure for them to break. And this allows us to do a form of multicolor imaging or we can have a mixed population of them and we can take an image. And then we can apply a pressure that's going to selectively collapse only one of these populations. And then we can take another image. And then we can apply the next higher pressure to collapse the next population, and so on. And from that, recover information about what are the different components that are present in the image. And actually, we can turn it into a linear algebra problem, for those of you who enjoy those, and really obtain quantitatively what are the different contributions of each of these things. And so right now, we have 3 different colors of ultrasound. While we're at it, since we're messing around with a protein that sits on the outside of these particles, we can functionalize it. So now if we want to modify how these things interact with the body or how they interact with different cells, we can just take this GVPC protein and we can paste some things at its end, okay, additional protein functionalities that can do things like change the electrical charge on the surface of this particle, which affects how it distributes through the body. Or we can put little protein signals on there that act as don't eat me signals for immune cells, okay, so the CD47 signal. Or we can purposely target them to cells. So here we've appended a little peptide sequence on the end that when it recognizes tumor cells, a particular protein on the surface of tumor cells, it causes the gas vesicles to dock to those cells specifically. Okay, so everything I've told you about so far is us just working with these proteins in isolation, okay, which was already pretty exciting. But the vision that I sold you on, hopefully, in the beginning was that we wanted to image cells. Okay, so now what we want is for the cells that we're interested in tracking around the body to produce these gas vesicles themselves based on a genetic sequence that we put into those cells. And as our first target, we became interested in imaging bacteria, specifically microbes in our guts. And this is really important because, as I'm sure most of you know, we have a really rich ecosystem of bacteria that are living in us. Okay? Something like 100 trillion cells. Okay, and every time you eat yogurt, you're eating like 1 billion more. Okay? I mean, of course, you're letting some of them out as well. And what's important is that a lot of what we know as scientists about what's in our guts actually comes from feces. Because people take the feces and they look at what microbes are there and try to infer what's happening in the gut. But that's not very representative because as people, including our own, my colleague here, Sarkis Mazmanian, have discovered, depending on where you are within the gastrointestinal tract, whether you're in the small intestine or the large intestine, you have completely different populations of species that are present there that are doing different things and are interacting differently with the cells that are part of our body. And so if we really want to understand our microbiome, we need to be able to see them within the context of the living, breathing organism and not just in things that come out of it. That's for basic science. But in addition, as I mentioned earlier, synthetic biologists have been engineering microbes to carry out new functions. And there's an entire new field of people doing this because we figured out really good ways to genetically manipulate them. So now we can have things like microbes that are going through our guts detect things like inflammation and release anti-inflammatory compounds, or detect the presence of a pathogen and kill that pathogen. There are even microbes that like to live in tumors. So people have injected microbes into animals that have cancer and had those microbes go and populate the tumor and destroy it. So there are lots of interesting applications. And again, in these cases, we want to see where are these microbes and what are they doing. And so when we started, we said, OK, let's just take the genetic program encoding these gas vesicles in cyanobacteria and just copy and paste it into E. coli, which is the good E. coli, of the kind that we wanted to image in the gut. And this turned out to be much more challenging than we thought. If you just take the genes and move them over, even if you try to optimize them a little bit so they're more compatible with the E. coli environment, we got nothing. Okay, this was very disappointing. Then we said, OK, maybe that's too much of a jump. Let's find the species that is the closest genetic relative of E. coli that has gas vesicle genes in its genome, and let's take those genes and move them over into E. coli. Okay? And when we did that, we started to see some gas vesicles. Okay? So this is an electron microscopy image of a whole E. coli cell, and all of these little white dots you have in there are little gas vesicles that now have been formed based on us putting these new genes into the E. coli. And we can also lyse these bacteria and look at these particles in isolation. And this was really exciting, and we thought we would see ultrasound contrast, but unfortunately, when we put these cells under ultrasound, now this is with live cells, again, we didn't really see anything, and this was disappointing. And we figured that this is because, although we're forming gas vesicles, there are these really small ones that are not really producing much contrast. Okay? So we want the ones that have the structural properties that are present in the cyanobacteria. And so then we looked a little bit more closely at these genes. And what's known is that some of these genes are the structural genes. They're the bricks that form the structure. And some of these other genes are accessory factors or assembly factors that are like the cranes that are putting these bricks in place. And we hypothesized that we could take the crane genes from this near relative and combine them with the structural genes, the bricks, from the cyanobacteria that form really nice gas vesicles, and get this whole kind of hybrid system to work in the E. coli if we put them together. And luckily, that turned out to work really well. So now when we have this hybrid gene cluster, we started to see more and larger gas vesicles formed inside of these cells. And for the first time, we could see live cells producing ultrasound contrast in our lab. It was a very exciting day. And then we could optimize this a little bit by adding this other protein I talked about earlier, GVPC, that sits on the outside and strengthens them. And now we form gas vesicles that really look like the ones that came from cyanobacteria. It's very nice. And we called this genetic construct ARG1, or acoustic reporter gene 1. And when we really crank up the expression in these cells, this is what it looks like. Okay? So all these little objects in here are little gas vesicles. It's a little bit exaggerated because transmission electron microscopy is doing like a 2D projection here, so we're not really filling the entire thing with them, but about 10 to 20%. And if you can crank this up enough, you can get the E. coli to float to the top. And so the kind of questions then we asked is this good enough for us to actually do some real imaging? So one test for that is to hook up these gas vesicle genes to a genetic circuit and see if we can follow the dynamics of that circuit. And we picked the simplest gene circuit possible, where we have a little sugar that we're giving to the bacteria. And when we give that sugar, it turns on the production of these genes. And normally that's an experiment that people would be doing with fluorescent proteins and looking at it optically. But now we're doing acoustic proteins and looking at it under ultrasound. And we can get this nice transfer function between our chemical input and our ultrasound output that people normally would have just done with fluorescence, but now we can do it acoustically. And of course, we want to know how sensitive is it. So currently, we can detect about 10 to the 8 cells per milliliter, which sounds like a lot, but actually it's not. It corresponds to a volume fraction that these cells occupy of less than 0.01% and is in line with the densities that these cells reach in some parts of our gastrointestinal tract. So we're doing pretty well. So then we wanted to put it in the body. And actually, the cell that I showed you earlier is a special form of E. coli called Missile 1917 that has been used for exactly 101 years, which is where this name comes from, as a probiotic. Okay? Because when you give these cells to somebody, they will colonize some of the gastrointestinal tract and exclude pathogens from settling in there. And this is not available, I don't think, in the US, but in Germany, you can buy it as Mutaflor and then sneak it in. Which is not what we do. We do it. So now we've taken our genes, we've put them into these E. coli, and we're going to image them in the gastrointestinal tract. And as an illustration of the advantages of ultrasound compared to optical imaging, we're putting them in this spatial arrangement within the colon, where on the outside we have E. coli that are expressing our ultrasound reporter genes, acoustic reporter genes. On the inside, we put in E. coli that are expressing light genes that will produce light. Okay? When we look at these by ultrasound, we can very clearly see where the ultrasound ones are, and this dark spot is where the light ones are. If we invert this arrangement, now the acoustic ones are on the inside, we see ultrasound contrast on the inside, and the outside is dark. And in both cases, when we look at this under luminescence, looking at light, it looks about the same. You just see a blob. Okay, and that's because with ultrasound, we can really get this very, very nice spatial precision deep inside, whereas with light, things get blurred. And we're doing a lot of other things with gas vesicles that, unfortunately, I don't have time to tell you about, but they involve brain imaging, looking at brain activity, and where these things can enhance it. We can see them under magnetic resonance. We're trying to put these genes into animal cells. And we got actually some very nice results in the lab about an hour ago on this. They have very interesting optical interactions, and some of these things are published. And then there's some other things that I'm not at liberty to discuss, thanks to my students. So, so that's talking about the imaging side of what we do. And now I'd like to switch gears a little bit and talk about the other side of the equation. Right, so now we see where these cells are in the body. Now we want to be able to tell them what to do based on their location. And for that, we're again going to turn to ultrasound, because in addition to being used for imaging, ultrasound energy can be focused inside the body and actually can be focused deep inside with millimeter spatial precision, okay? And then it can deliver energy. And this is used clinically for non-invasive surgery, because if you deposit enough of this energy, you can heat things up to the point where you're ablating or frying the tissue. And so it's a way, for example, to take care of prostate tumors, of uterine fibroids, and even some things in the brain, as we'll see a little bit later. Now, in our case, we want to talk to the cells. We don't want to fry them. And so what we're going to do is apply the ultrasound, but have it elevate the temperature by just a few degrees Celsius, where between 37 and 42 degrees, most cells and tissues can tolerate it just fine. And what we want is for this small change in temperature to act as a signal that our engineered cells can perceive and then do something based on that information. Okay? And so for that, we want to put in some kind of genetic program into these cells that has a temperature sensor that can then turn on some functionality that we want these cells to carry out. And so to do that, we again turn to nature. And actually, a lot of cells already have the ability to sense temperature. Just like we know when we're having a fever, and it turns out some bacteria also want to know if you're having a fever so they can infect you better under those conditions. And all of our cells have these things called heat shock proteins that get turned up when it's hot so they can help prevent proteins from misfolding and those kind of things. And so we looked at what's available in nature and tested them. And we basically looked at the performance of this gene circuit that's driven by these temperature sensors under cold condition and under hot condition, and said, how much of a change is there in the expression of these genes? And most of these switches didn't perform very well. They had very mild changes, but we had two of them that performed beautifully. So if we look at them in more detail, we have these two genetic switches that, as a function of temperature here, now we're looking at gene expression, in this case with a fluorescent protein. We see that they have this very sharp switch-like behavior, where over the span of about 3 degrees Celsius for each of them, you have more than 300-fold changes in gene expression. Okay? And the way these things work is that in both of their cases, there are these proteins that bind to each other when it's cold, and they bind to the DNA, and they prevent that DNA from being transcribed and translated into the protein that's downstream of it. But when you heat them up, they unbind, come off the DNA, and allow the gene expression to take place. And so this was really great. We were very happy to have these switches. But there was a catch, which is if you look carefully at the temperatures where they're switching, they're both wrong. So one of them, this orange one, is switching at about too low a temperature, right? It starts turning on at around 38, 39 degrees. And this green one is turning on when it's too high, 42. And we don't want to heat the tissues to such an extent. And so now we had to, again, put on our hat as protein engineers and tune these switches. And one way to do that is using a technique that was invented by my colleague, Francis Arnold, that some of you may know, where we can make random mutations in the protein sequence, and we can have a bunch of these little bacterial colonies that each contain a different mutant, and we can do a screen to look for ones that have the new property that we want. And in this case, we can have a replica where we have two identical plates with the same colonies on them. We can grow one of these replica plates at the cold temperature, where we want the circuit to be off, and grow its partner at a hotter temperature, where we want the circuit to be on, and look for bacterial colonies that are dark here and light here. Okay? And when we do that, because we've introduced random mutations, most of these guys, we've ruined them. Right? We have a lot that are just already on at the low temperature, for example. But there are a few, when we do that, that have the behavior we want, like this colony here. And when we perform this screen at a few different temperatures, we can generate now an entire library of thermal switches. Okay, where here this is showing 6 different ones, where they all have this nice switch-like behavior, but we've tuned them like a thermostat, where you tell me where you want the switching to occur, and I can give you the gene that allows the cells to do that. And because we started with 2 of them, we can actually build some more complex genetic circuits. For example, we can have 2 different things turning on at 2 different temperatures. Okay? Or we can have a bandpass filter, meaning that we have something that's only going to turn on between temperature A and temperature B. And then, of course, we want to see if we can control this with ultrasound. And so the way we do this in some of our experiments is that we have now a lawn of these bacteria that contain this genetic circuit where when you reach 1 temperature, you're going to have green expression. And when you reach an even higher temperature, you start to see red expression. And we'll put a piece of tofu on top of it because it simulates the acoustic properties of tissue. And then we apply focused ultrasound through this tofu at this bacterial plate. And we can do this under the guidance of magnetic resonance imaging, which in real time tells us exactly how high the temperature is getting, so we can turn up or down the ultrasound to dial in exactly the thermal pattern we want. And then we can look a little bit later at the gene expression pattern that we've generated. And so here, using ultrasound through a piece of material that you wouldn't be able to shine light through, we were able to apply the ultrasound with millimeter precision and paint this bullseye of gene expression, okay, with millimeter scale. And then, of course, we want to do this in vivo. And so as a quick demonstration, we injected bacteria in a few different locations in a mouse. And then we apply ultrasound only to this right hind limb. And then we can see the gene expression turning on just at that location. Now, you know that we take inspiration from Nobel laureates, as I mentioned earlier. But we also take inspiration from Caltech undergraduates, because they're just as brilliant. And one of their most brilliant contributions occurred at the MIT campus preview weekend in 2014, where a bunch of Caltech students dressed as MIT students came to campus, and you guys probably have heard of this, and there were all these excited new MIT admits, students and parents walking around, and these enthusiastic MIT students were handing out free MIT mugs to everybody. And all these students and parents were super excited, and they took them home. And then as soon as they put any kind of hot liquid in it, it turned from saying MIT to saying Caltech, the hotter Institute of Technology. And so we were inspired by this and said, hey, we have something that switches with temperature. And so we made these bacterial plates that contain 2 different kinds of bacteria in a pattern, where one of them says MIT and the other one says Caltech. And if you take this bacterial plate and you incubate it at a temperature of 40 degrees, at the low temperature, it says MIT. But then if you take these bacteria and you put them in the high temperature, now it says Caltech. And these are available for sale after the show for a few $100,000. Okay. So let's see how we're doing. So I think I have a few minutes. So I want to tell you about one additional story, just in 5 minutes or so, if you'll indulge me, where everything I've told you about so far is how we implement our ideas and technologies in bacteria, which are a very natural starting point, because it's easier for us to engineer them. They're important in the gut and other places. But we're also interested in doing these kind of things in other tissues and cell types. And in particular, I want to tell you about something we're doing in the brain. Okay? Because we want to also be able to alter the function of neural circuits that go awry during different kinds of diseases. So if we think about diseases like epilepsy or depression or addiction, from what neuroscientists understand about them, many of them are not diseases of cells that are dying somewhere, but they're more diseases of neural circuits, okay, where there's some location in the brain that has a certain type of cell, of neuron, that is malfunctioning. You know, maybe it's overly excited, or it's not as excited, or it's signaling to the wrong other cell types. And unfortunately, our current treatments for these diseases are inadequate, because we take drugs that don't just go to those cells in that circuit, but they go everywhere in our brain. And that's why we have these terrible side effects, or are not able to use them at really efficacious doses. Or we have surgery, where we have to go in and cut out a piece of the brain, or implant an electrical stimulator, and those kind of things. And we'd like to go beyond that using some of the technologies that we're developing. And so the task is to develop a technology to treat brain diseases that is non-invasive, doesn't require surgery, but can give you a combination of location, specificity, cell type specificity, so that at that location, out of the different types of neurons that are there, we can manipulate just specific ones, and the timing and dose of when that happens. And so we developed this technology that we call ATTAC, or acoustically targeted chemogenetics, which I'll explain what that means in a second, that achieves this by combining, no surprise, ultrasound as the energy that we can focus to a certain location in the brain, and genetic vectors using viruses that we've co-opted that can deliver not their genes, but genes of our choice to that location where we're applying the ultrasound. So it gives us the cell type specificity because these viruses will express the genes only in a subset of cells at that location. And then the timing comes from the fact that the gene that we're putting into these viruses encodes special receptors that now, when they're produced in these neurons, allow them to be controlled with this designer drug that itself doesn't do anything else in the body, but only acts on that receptor as a lock and key. Okay, and that can be used to turn those neurons on and off. So I'm just going to give you a little bit more information on these things. So ultrasound, as I mentioned already, can be focused at depth in tissues, and it can be done in the brain as well. And so there's an FDA-approved treatment for essential tremor, where there's under MRI guidance, ultrasound is applied, where it gets focused on this millimeter scale location within the brain, and in this case, again, it's ablating something, so it's heating it up and breaking it, and we don't want to do that. What we want to do instead is apply ultrasound to a certain part of the brain and allow viruses that we're injecting to only penetrate into the brain at that location. And the reason that works is that normally our brain has what's called the blood-brain barrier, which prevents things from the bloodstream from getting out into the brain itself. It's a good protective mechanism. But what we're doing here is that we're injecting some bubbles into the bloodstream, where when the ultrasound hits these bubbles, it causes them to gently expand and contract, and essentially massage the blood vessels from the inside, which causes them to open up their junctions and allow molecules, including small viral particles, to get out, and that stays open for about 2 hours. Okay, so if in that 2-hour window after you apply the ultrasound just at that location, you inject some of these viral particles, then they'll be able to come out and infect the neurons. Okay, and the virus that we're using for this is called AAV, or adeno-associated virus, which was approved by the FDA last year as the first gene therapy in the United States for eye disease. And the key to this is that, viruses sound scary, but what we're doing here is we're changing the virus so that instead of delivering into the cell its own genome, which it would use to make more copies of itself and then proliferate around and kill the cells, instead of doing that, it's delivering genes that we've chosen. So this virus is not capable of replicating, but it's just acting as our agent, just like the cells were earlier. So it's going to deliver these genes, and these genes have an address tag that says at that location, just express my genes in the excitatory neurons, for example. And so then we need to talk about what gene we're using. And the gene we're using is these things called chemo-genetic receptors that my colleague Henry Lester here was one of the first people to develop, where the idea is that normally neurons have these receptors that when neurotransmitters act on them, causes these receptors to either excite or inhibit the neuron, depending on what receptor we're talking about. But with chemogenetics, what we've done is taken these receptors and again genetically modified them so that instead of responding to neurotransmitters, now they will respond to a designer drug of our choice, okay, that we can inject into the system. So that whenever we administer this drug, this new designer receptor will be bound by this drug, and now it'll excite or inhibit the neurons. So whatever neuron we can deliver this gene to that encodes this chemogenetic receptor now can come under our control. And the drug we're using, this molecule CNO, which is a metabolite of an FDA-approved substance. And so the idea is that you can have a brief ultrasound session, okay, where you can apply the ultrasound to a certain part of the brain that will specify what location we want to control. Okay? Then we inject this viral vector that's going to deliver these chemogenetic receptors just to that location. Okay? Then they'll be expressed there. Then you don't have to do ultrasound anymore again. And now, whenever you want to turn on or off the neurons at that location, all you have to do is just give this drug. Okay? And so now this combines spatial, genetic, and temporal specificity in a convenient way. And so we've implemented this in mice as a starting point. And what we're doing here is targeting a brain region in these mice called the hippocampus, which is important in the formation of memories. And we can watch by MRI where we're opening the blood-brain barrier. So you can see here that we've successfully targeted this brain region. And then later we can look and see if we were able to successfully deliver the genes into this region using fluorescence. And so here we've beautifully gotten our gene into the neurons that we wanted. And as a proof of concept of whether this can actually do something in the brain, we've implemented in this model of learning called fear conditioning, where we take a mouse and we put it into this chamber and we administer mild electric currents, okay, kind of like licking a 9-volt battery that's unpleasant to the mice. And they remember this and they don't like it. So that if you take these mice and you bring them into the same environment the next day, they have this freezing behavior where they're like, oh man, am I gonna get this 9-volt battery thing happening again? That's only if they can remember what happened to them. Okay? And so what we're doing here is, going back here, we're in the hippocampus, which is needed for forming these memories. We're expressing this chemogenetic receptor that we targeted by ultrasound that's going to turn off those neurons whenever we administer this compound CNO. And so if we administer this compound before doing this training, then the mice should not be able to form a memory of it. And indeed, that turned out to be the case. So now, pharmacologically, we can control the activity of a specific part of the brain. I'm pretty excited about it. Okay, so to summarize, what I hope I've been able to convey to you is that one of the future directions of medicine involves having little microscale devices, just like the Fantastic Voyage, that are going to roam around our bodies and are going to diagnose and treat disease. And that some of these devices, instead of being electromechanical things that are shrunken down, are going to be cells that we're going to engineer to do our bidding. And in our lab, we're trying to stay in touch with these cells, okay, by developing technologies that allow us to see where they are and what they're doing and be able to send them commands to image and control them. And I was able to tell you about some of the work we're doing. If you're interested in learning more, there are a few papers that you can look up on our lab website or come and ask me. And of course, really, I'm just a spokesperson presenting the work of this amazing group of people that we have in my laboratory. I think the kind of research that we do is really uniquely possible at Caltech because we're combining biology and molecular engineering, and we're combining physics and finite element modeling and nuclear spins and sound waves. And it really requires a team of people that have very diverse backgrounds to come together and do this kind of research. And so in my lab, my home base is in chemical engineering. Right? I'm a chemical engineer. But the students in my lab come from chemical engineering, from medical engineering, from neuroscience, from chemistry. The postdoctoral fellows in my lab come from physics backgrounds. And so we're able to combine all these ideas in a unique way because here at Caltech, we're small and we don't have barriers that keep these disciplines apart from each other. In addition, we really benefit from support. So we're lucky to get a lot of support from the federal government and various external foundations, but we've also had some very generous support from the Caltech community in terms of grants from the Jacobs Institute, the Center for Environmental and Microbial Interactions, which supports a wide variety of microbial research at Caltech, the Rosen Bioengineering Center, and the Heritage Medical Research Institute, which supports some work that's translationally oriented. So with that, I am really grateful to you for your wonderful attention, for laughing at some of my corny jokes, and I would be happy to take a few questions. Thank you very much.